Write down. Yes. So now, the main function is this. So white blood corpuscles or leukocytes, do they have respiratory pigment? Do they have respiratory pigment?
Mom, the screen is not visible.
Is it screen not visible?
No, ma'am.
Is it will all? Nobody can see the screen? Okay, just give
Mom,
me
it's
2
visible.
minutes. It's visible?
Ma'am,
It's
now it's
visible.
visible.
Kavya, is it visible? Malaika, is it visible?
Yes, ma'am. Now it's visible.
Okay, fine. Yeah. Do let me know if you are not able to see. Okay. Next. Function of leukocytes, as I said, the shape of the first is the shape. Shape is irregular and polymorphic nucleus. Size, curvy portion for test. What we have completed in the last class, last test, what was included? Biotechnology, right? So this time it will be application part. Okay. Actually, it will be both biotechnology and application part. I'll give some weightage on the initial chapter, and also I'll give more weightage for the second part of biotechnology. Clear? 3 to 20 micron lifespan, 3 to 30 days. Total leukocyte count TLC is 6,000 to 10,000 mmQ. RBC is to WBC. The ratio is 600 is to 1. If you find 600 RBC, you see only 1 WBC there. Increase in TLC is leukocytosis. Decrease in TLC is leukopenia. This condition is seen in infection, any kind of infection, and this condition is seen in AIDS, typhoid diseases. Right now. Please mute yourself.
Ma'am, I can't see your screen.
Nobody can see the screen.
No,
No,
ma'am.
ma'am.
You should tell me.
No,
now.
ma'am.
Okay, let me.
Ma'am,
Yes,
now
ma'am.
it's visible.
Now it is.
Now I've stopped. When you say now it's visible, I stopped. Just wait. Share the screen. Do let me know if you can see the screen. Okay, just a minute.
Yeah, ma'am, visible.
Visible.
Visible? See ya.
Mom, it's visible.
Yes, ma'am. It's visible.
Okay.
Ma'am, what is polymorphic nucleus?
Polymorphic means the nucleus is not same shape. It is of different shape.
Okay, ma'am.
I'll send you on. Just wait. Can you all see the screen?
Yes, ma'am.
Done? Are you all done? Next. So this is how the leukocytes look. Can you see the leukocytes? Next. See, there are different types of leukocytes. First thing. What happened, Shantanu? You cannot see the screen. Shantanu, what happened? Can you all see the screen? Yes or no? Okay. Now, so basically leukocytes have granules. What do they have? Granules. These granules take up the stain. Some leukocytes have granules, some leukocytes do not have granules. So they are categorized under agranular and granular leukocytes. There is a difference between agranular and granular leukocytes. Granular and agranular leukocytes. Is this point clear? Now, next. Granular leukocytes, where in the cytoplasm there is a granules. Agranular, there is absence of granules. Under granular, there are three different types: basophil, neutrophil, and eosinophil. Under agranular, you find lymphocytes and monocytes. Lymphocytes and monocytes. Now, okay, let me draw it here. First, granulocytes of WBC, the percentage is around 70%. Granulocytes you found is 70%. Under this, you come across neutrophil. Next, you have basophil. Next, you have acidophil. Done? Next, neutrophils, first thing is their maximum in number. Maximum in number. Can you all see the screen?
Yes, ma'am.
Okay. Maximum in number. Next, that is around 60 to 65%. They are multi. Yeah, what happened to this now? Maximum in number around 60 to 65%. Multi-lobed nucleus. The main function is phagocytosis. They are phagocytic cells. They help in defense mechanism. So they are phagocytic cells. Next. Basophils. Minimum. The number is minimum. Around 0.5 to 1%. They are trilobed nucleus. They have trilobed S or U-shaped nucleus. What is the function? They secrete histamine, serotonin, this is a function of basophil. Next, you have acidophil. Acidophil is also called eosinophil. 2 to 5% bilobed nucleus. The main function is they are antihistamine. Antihistamine. They secrete histamine, but these are antihistamine, and they also take part in anti-helminthic effect. Wherever there is an infection due to helminths or worms, acidophils gets activated. Please write down. this now. Hello, are you done with this? Hello. Okay. Can you hear me?
Yes, ma'am.
Now, so neutrophils is about 60 to 70%. Okay, we'll come back to the other. Yeah, after acidophil, after this all are granulocytes. So when we discuss about a granulocyte, they are about 30%, which consist of lymphocytes and monocytes. Lymphocytes around 25% and monocytes around 5%. They are the second maximum in number. Form, very important line, form third line of defense. Do you remember the third line of body defense? First line, second line, third line. Do you remember this? We discussed this. Which are the two lymphocytes? Which are the two lymphocytes? B and T lymphocytes. Which among these B and T lymphocytes produces antibodies? Which among this produces the antibodies? B lymphocytes. Very good. What is the function of T lymphocytes? They attack direct cell and they kill, right? So now whenever B lymphocytes, whenever monocytes, okay, whenever monocytes enter into tissue, what they become? They become what? Very good. They become macrophage, right? Then, where does B and T lymphocytes form? Where does the B and T lymphocytes form? Very good. Bone marrow. Where do you think the B lymphocytes get mature? B lymphocytes get mature. B and T form, that's a bone marrow, and T mature in thymus. Do you all remember these parts? Yes. Yes, very good. Any difficulties to anyone? Gauravi, yes, it is thymus. Is this part done? Next. After this, we'll be discussing about platelets. What is the other name for platelets, my dear students? Thrombocytes. Who can tell me the main function of thrombocyte? Okay, before we getting into platelets, an easy way to remember, never let monkeys eat banana. You can keep any mnemonics, whichever you like. Never let monkeys eat banana. Neutrophils, lymphocytes, monocyte, eosinophils, basophils. What is that? Ma'am, preventing. Okay. Yeah, they prevent the clotting, right? Neutrophils. So I think this all. You can just see how the phagocytosis takes place. Can you see the slide of phagocytes? How the first is phagocytosis of foreign particle. Then lysosome fuse with the fuse. Then after that they degrade and they degrade. Okay, the foreign particles degrade. After the degrade, they are after the degrade these are removed. So these all things happens with the phagosomes. Did you got the point? How the neutrophils work? With the help of lysosomes, the phagosomes will mix to form the phagolysosomes. They degrade the antigen. And now, what is this MHC? MHC is major histocompatibility. Compactability. What does it mean? Mean the antigen is presented on the surface of the cell so that this antigen is recognized by B cells and produce antibodies. It's a signal to B cells saying that somebody has entered in our body and we need to protect by producing the antibodies. It's a signal. Who present it? MHC present it. On what? On the cell surface of the any cell. Macrophages or neutrophils, any cell. Got the point? Next. Eosinophil, See, everything is given. Development is not important. Lifespan is also not important. The important thing I have told you. Basophils. We have discussed. Lymphocytes. You can write down about the lymphocyte if you have missed something. Can you see the lymphocyte slide? Can you all see the lymphocyte slide?
Yes, ma'am.
Okay, write down about the lymphocytes. I think everything is mentioned: B lymphocyte, T lymphocytes, direct attack antibodies. This is about the monocytes. Monocytes, phagocytosis develop into macrophages in tissue. All these points is been mentioned. Next, we are going to okay, leukocytes. Still, we need to discuss few points about the leukocytes. Leukocytes have few important see neutrophils basically, which is a leukocyte, first reach to the site of infection. Got the point? WBCs are the one that reach to the neutrophils are the one that reach to the site wherever there is an infection, the first cell to reach. This point is clear. Now, how does it happen? One is margination, other is diapedesis, and the third step is chemotaxis. The infected area outside the blood vessel by three sites. The leukocyte reaches the infected area. Can't see the screen. Now we can see, Kavya. Can you see it now, Kavya? Let me check. Relog in my system. Okay. Can see? Okay, fine. Today can see, cannot see. That's what is going on. Okay. So infected area, when the infection is outside, how do they reach? They reach it margination, diapedesis, or by chemotaxis. These are the three methods how they reach to the infected area, because leukocytes are the cells that reach to the infection area. Okay. Now, margination is where, what is margination? Let us discuss. Margination is slow down. See, in the blood vessels, the blood is flowing. Along with the blood, there is RBC is flowing, WBC is flowing. They need to get slowed down. How do they realize there is an infection? So what they happen in margination, they slow down by cell adhesion molecule secreted by endothelium. Secreted by endothelium. Is this point clear? This is called margination. They become, when they are moving in the blood vessels, they just become slows down. Next, this is called margination. Write down. Okay, next. Next is diapedes. Diapedes means squeezing. If you have small space, you try to squeeze or come out to it. If you can see, the process of diapedes is started here. Here, where it's trying to squeeze out, and it is almost out here. So this process is called diapedesis. This is an important MCQ question. The squeezing of the leukocytes from the blood capillaries. Write down. Done? Everybody done? Can we move next? Okay. Chemo taxes. Chemo taxes, they secretes gathering in large number at area of tissue damage and infection by following the chemical trail of the molecules released by damaged cells. Like, the whatever the damaged cells, a kind of infection that is formed. Okay, here the infection, the tissue is getting damaged. Whatever the release content of this cell into this, they trace through the chemotaxis. That is called chemotaxis. When from where that uh, chemical is released, or from where that thing is released, from which cell. So let me track to that cell. That is called chemotaxis. Go through it and let me know if you have any difficulties with this. Is it done? Have you all understood the three basic ways in which the leukocytes reach to the inflammation or the inflammatory site or the infection site? Everybody understood? Just go through those three sites. Why are they slowed down at the start? See, because they need to, there's if something that is formed here, okay, margination, they cannot just keep on going. They need to slow down so that they can squeeze out. Abhita, got the point? They cannot just keep on moving. They need to slow down so that they can slow down and then they can squeeze out. If they keep on going with the flow, they will not reach to the infection site. Okay. Any other doubts? Now, phagocytosis, I have told you, the process of destroying the foreign substances or dead cells is called phagocytosis. Okay. This we have discussed. Now, platelets. See, what are platelets is important. You know, it is also called thrombocytes. But you all said the main function is for the clot. Very good. Now, let us read more about what is platelet. Platelet is not a cell, but they are the cytoplasmic fragments. Very important question. Fragments of megakaryocytes. Important MCQ. See, this is one megakaryocytes. The fragments from this cell is called platelets. The normal count is, write down, 1 lakh, 1.5 lakh to 3.5 lakh per mm cube is the normal count of platelets. Lifespan 7 to 10 days. RBC is to WBC is to platelet. What is the ratio? What's the ratio of the RBC and WBC? What's the ratio of RBC and WBC? (600 is to 1 is to 20). Next. Increase 20 is the platelet count. Okay, the ratio of RBC, WBC and platelet is 600 is to 1 is to 20. Increase in platelet count is thrombocytosis. Decrease in platelet count is thrombocytopenia. Do let me know once done. If you have doubts, you can ask me. Done? Hello. If it's done, say yes. Otherwise, say, ma'am, wait. Please, but do response. Done? Now, function of platelets. Function of platelets. They secretes vasoconstrictor that causes a vascular spasm in the broken vessels. They secretes a vasoconstrictor so that the vascular spasm they can in the broken vessels. The squeezing. This is the first function. Okay. Write down the first function of the platelets. The second function is form a temporary platelet flux to stop the bleeding. It forms a temporary, they help in the clot formation, but before that they stop the bleeding. Somehow they come to the site and they stop the bleeding. This is the second function. Next, secrete a growth factor that stimulate mitosis in the fibroblast and the smooth muscles and help in the lining of blood vessels. So what they do, they secrete some kind of growth factors so that they stimulate and divide the fibroblast cells and the smooth muscles so that they maintain the blood lining. Next, coagulation. The platelets take part in coagulation. How? How many factors are involved in blood clot coagulation? Tell me. Do you all remember we studied the blood coagulation factors? How many? Very good, Shantanu. Are they active in the blood or inactive in the blood? Are they very good, Abhida? Are they active in the blood or inactive in the blood? Inactive. When do they get active? When do they get active? When do they get active? Hmm? When do they get active? Nobody knows? Listen to me. Damage to blood vessels. Very good. Listen. Everybody listen. In the blood, you have 13 factors. How many? 13 factors. Those are the proteins. These 13 proteins need to get, for example, A, B, C, D, E, like that. You have 13 factors. These factors remain inactive in the blood because if they get active, there will be a formation of clot. So they remain inactive in the blood. How do they get active? If there is a damage to the blood vessels, A will get active. After once A is active, it activate the B. B will activate the C. C will activate the D, and D will activate the E. It keeps on going. Are you understanding? So the clotting, it goes in a cascade manner. One will activate the other will activate the fourth, like that. It keeps on going. So this is called the activation process. There are 13 factors that activate one after the other to form the clot. This is called clotting. It's called a cascade mechanism. Okay, activating one after the other. So when do they get active? They get active when there is a broken blood vessels, and in some cases when there is a slow blood flow, but basically they get active during the broken or damage to the blood vessels. Next, the sequential, can you see the slide? The sequential activation reaction cascade. I told you the activation is how A will get activate, A will activate the B, B will activate the C. So it keeps on a sequential of the clotting. The factor finally leads to the formation of fibrin. Fibrin means a clot. Finally forming a clot. Once a clot is formed with the fibrin, bloods are trapped in the fibrin mesh to form the hard clot. Once you have the fibrin clot which is formed, along with that, the also cells that have been trapped along with this network. Now the clot is formed. So, are you all done with the writing? Can I move ahead? Hmm? Have you all written? Are you all there? Yeah, you are there. Can you hear me? Can you all hear me? Gauravi, can you hear me?
Yes, ma'am.
Okay. Have you written this part? Done? So next. Coagulation disorders. There are few coagulation disorders. One is thrombosis, other is thrombus. Ma'am, wait. Done. Okay, Hiba will come back to the slide if you have missed something. So let us discuss about the coagulation disorder. Can you see the slide of coagulation disorder now? Thrombosis, thrombus, embolus, embolism, infraction. What is thrombosis? Is abnormal clotting of blood in unbroken vessels. Abnormal, like doesn't require to clot for so long, but it keep on clotting. That is called abnormal clotting. That is called thrombus. This thrombosis. Thrombus is when after a clot is formed, after the clot is clot should form where there's a broken vessels. But if the vessel is not broken, still it's forming a clot. This clot get detaches and comes in the blood, and that is called that attaches to the wall of the clot vessel. This is called thrombosis. So you have a clot which is forming. This condition is called thrombus when you have a clot attached to the blood vessels. When the same clot detaches and flows in the blood, it is called embolus. You can read. Embolus is a clot that comes off the wall of the blood vessels and travel in the bloodstream. Then, as it keeps on going in the bloodstream, this embolus, somewhere it clogs. That condition is called embolism. Is a blockage of blood flow by embolus that lodges in the small blood vessels. That is called embolism. Infraction, you all know, we have studied, refers to cell death that result from because here it's a block. So the cells surrounding this area are dead. The infraction is responsible for the stroke and heart attack. Please write down this. If you have any doubts, you can ask me. See, it's not platelet that lose, but the amount is important. As I said, decrease in the clot is also a thrombocytopenia and thrombocytosis that will take place. Okay, as we come across or prone to a number of disease as we get age. It depends on different conditions. Is it clear? The number that will tell you whether the clotting is happening or not. If it's too much, it leads to clots. If it's less, also it's a problem. If it's too, also it's a problem. Too low is also a problem. Is it clear, Shantanu? Drumsticks.
Mom, the screen isn't visible.
Now it's visible.
Yes, ma'am.
Okay. Why are you sad? Why are you sounding so low? Hmm?
I don't know, ma'am.
Okay. Please write it down quickly. Done? Acha, acha, acha. Okay, I got it. So it does. Yeah. Hello, is it done? Are you all there? Hmm? Okay. So, Iba, which one you want me to show? Which slide? Okay, fine. Okay. So, you got the slide, Hiba?
Yes, ma'am.
You got very too much of internal roots, huh? To get the things. Hmm?
No, ma'am.
No, that's really nice. You need to help each other. I always tell my students, you can raise yourself only when you raise others, right? Okay, that's really nice that you are helping each other. So where we? Okay, are you all done? Now quickly go through WBC, sorry, RBC, WBC, and platelets before we move to. Can you see my summary here? Okay, no. Let me finish this also. Can you see the summary? What I have made with the WBC, RBC, and platelets. I just need to look into this, and many of the things I'll be able to answer from here. And also, serum. What is serum? Yellow straw colored fluid. See, whenever there's a clot, you get a clot. Around that, you find a serum. Important question is, can serum clot? What would be your answer? Can serum clot? Can serum clot? Very good, Lena. Serum cannot clot. You can see here I have drawn this clot in the red color, which I made it black now. Surrounding to that, there is a serum, yellow color. They cannot clot because it doesn't have fibrinogen protein. Very important question. If somebody asks you MCQ question, okay, this is an MCQ question. Many times this question is asked. Can serum clot? No. Why? Because they do not have fibrinogen. Next, what is lymph? The other vascular tissue is lymph. Lymph is a colorless fluid without RBC, WBC, but they have, sorry, RBC and platelets, but they have WBC. You know, B lymphocyte, T lymphocytes are present in the lymph. So there is a colorless fluid without RBC and the platelets. Only WBC is present. They have only WBC. It's a middleman between the tissue fluid and the blood. And very important function of lymph is they help in absorption of fat and fat soluble vitamins. You know that the nutrients, the food what we eat will get simpler. After it gets simpler, what happened? They reach to the site. Okay, they reach to the site by the help of a blood. The carbohydrates, proteins, whatever we eat, the nutrients reach to the site by the through the blood. But fat and fat soluble vitamin will not reach. They can only reach through lymph. So this is an important function. So what are the vessels that are present in the intestine which absorbs the lymph, which absorbs this fat? What are they called? Who can answer this? I said in the intestine there are blood vessels. Along with the blood vessels, there are lymph vessels that absorbs the fat. What are their lymph vessels called? Very good, Abida. It is lacteals. Do you remember Hiba? Very good. Lacteals. Pearl. Very good. Okay. Now I have to say good to all. So it is lacteal. Is it clear? Shantanu? Done? It's a lacteal. Any doubts? Any doubts to anyone? Okay. Gauravi, no doubts. Okay, good. What about others? Quickly tell me before we move to the next, that is muscles. So, so, so we have done with the connective tissue. We are done with the epithelial tissue. We're done with the connective tissue. Very, very important part of your exam. I want each and everybody to refer to NCRT book along with my notebook, and I want you all to make a summary of epithelial and connective tissue and send me. Email me, please. Okay. What did I ask you? You all are going to refer to the NCRT book refer to my notes, make a summary, draw the diagram. Summary means not required that you need to sit and write. I'm not allowing you to write. Don't sit and write. But I mean, no, make a flow chart, make a diagram, explain the diagram, make it simpler and easy. Is it point clear? Will you all do this? Epithelial tissue and connective 110% question from this part. Which email? Actually, you do not have my email ID. Write it down. Small letter, okay? Yes, it's small. You can email me your summaries on this, because if you send through Teams or if you send through WhatsApp, it's difficult. I have to delete. So I don't want to delete your hard work. So send me through email. Is it clear? Refer NCRT and make summary. Refer my notes and make summary. Then solve the MCQ questions. 110% question will be from this part. I'm telling you. Then don't tell me, ma'am, I lost marks in not answering from epithelial or connective tissue. Okay, next. The next connective tissue is muscle. Write down muscle. The origin is mesodermal in origin. M for muscle, M for mesoderm. How many muscles in your body? 639. What is the main property? Excitability and contract. Do they get excited? Yes. Do they get contract? Yes. Then only we move, right? So this is an important thing that you should know. Study of muscles is called myology or sarcology. Please write down. Study of muscles is called myology or sarcology. Done? Any doubts, my dear students? Tanisha, how are you studying, Tanisha? Tanisha. See, my dear students, I want each and every one to score full scoring in all the subjects, yes. But don't give me any, I don't want anyone to get unsatisfied after coming from the examination, say, ma'am, we couldn't say this. No, no, no. You have time. Please put your efforts. Please put your efforts. And this is a time where you need to put your efforts. This year is very critical. I want you all to put your efforts and all your dreams come true. But for this, your hard work is important. Your sincerity, your hard work, and you're focused. See, today, I don't know, many of them are not coming to the class. How many? There are 30 plus something. This is not going to help. If you're punctual, if you're focused, you should be from the day one. Otherwise, don't attend the first class also, right? If you really want to score well, you have to be very sincere. Anyway, it's structure of muscle fibers. We need to do structure of muscle fibers. Do we have time? Basic thing, I'll let you know, is a long, cylindrical, and unbranched. You have long, cylindrical shape, and unbranched means it is not showing any kind of branching. This structure of muscle fiber has myofibrils. So what are myofibrils? These are the proteinaceous rod-like structure present in sarcoplasm. Sarcoplasm is a cytoplasm of the muscle. What is sarcoplasm? Cytoplasm of muscles. In the cytoplasm, you get protein fibers, rod-like structure called myofibrils. These myofibrils are made up of two myofilaments. One is thick and long myosin, other is thin and short actin. You have two myofilaments in the myofibrils, and this myofibrils is present in the sarcoplasm. Please write down structure of muscle fiber is a long, cylindrical and unbranched. Myofibrils, these are the proteinaceous rod-like structure present in the sarcoplasm. Next, myofibrils are made up of two filaments. One is actin, other is myosin. Got the point? Did you all got the point? Hello, are you all there?
Yes, ma'am.
Okay, next. So if you see here, This is a the outer covering of this is called sarcolemma. The plasma membrane of the muscle fiber is called sarcolemma. The cytoplasm is called sarcoplasm. And this sarcoplasm has myofilaments, myofibrils, and this myofibrils have myofilaments. So first thing is outer is sarcolemma. Inside you get sarcoplasm, that is cytoplasm. In that cytoplasm you find is myofibrils. This myofibril is made up of myofilaments. Which are the two myofilaments? Tell me, which are the two myofilaments? Myosin and actin. Very good. Myosin and actin. Is it point clear? Is it point clear? Myosin and actin. The new students, are you getting the point? So you have first, you have sarcolemma. In the cytoplasm, you have sarcoplasm. Sarcoplasm has myofilaments that are two types, actin and myosin. Is this point clear? So the thick is myosin and the thin is actin. The thick filament is myosin and thin is actin. The thick is myosin, the thin is actin. This myosin, we need to draw the structure. Okay, we'll come back to that point. So this is how the myofilaments are attached. The myofilaments present in the muscle fibers, which are those actin and myosin. Next is we need to discuss, okay, structure of muscle fibers. The outer layer is called sarcolemma. The cytoplasm is called sarcoplasm. Okay. They are endoplasmic. ER is present. ER is endoplasmic reticulum. The endoplasmic reticulum of muscle is called sarcoplasmic reticulum, which stores calcium ions. Very, very important MCQ. Which ions do they store? Calcium ions. Then the plasma membrane or the sarcolemma of muscle fibers invaginate to form T tubules. They invaginate to form what? T tubules. Are you getting the point? They get inside to form T tubules. Then you have mitochondria of muscle fiber called sarcosomes. Sarcosomes are the mitochondria. Sarcoplasmic reticulum is the ER. The membrane is called sarcolemma. Inside the sarcolemma, there is invagination that is called T tubules. There are myofibrils, there are actin and myosin. And the nucleus is syncytium, means it is fused or it is to the corner, it is to the periphery. Then they have a glycogen granules. They have a phosphokeratin for the energy. So they have one is a glycogen granules that provide energy, phosphokeratin that provide energy, and they also have a myoglobin. What is the function of myoglobin? Quickly tell me. What's the function of myoglobin? Come on. Oxygen binding, to carry oxygen. How many oxygen molecule does the myoglobin carry? No, not energy. Oxygen, very good. How many, ma'am, have you finished the biomolecule chapter? It's not from here. Yeah. So, how many oxygen molecules do they carry? Anyone? One oxygen molecule. Yes, one oxygen molecule. See, hemoglobin is carried in the blood, okay, whereas in the tissue or in the muscle, not in the tissue, basically in the muscle, oxygen is taken care by myoglobin that carries one oxygen. Please write down. If you have doubts, you can ask me. Please write down, and if you have doubts, you can ask me. Clear? Any doubts to anyone? Done? Are you all done writing this? No doubts? Tell me. No doubts. So can we log off if there are no doubts? What about others? Everything clear? Okay then, bye bye. Take care. Keep learning. Bye bye, everyone. Take care. And do the summary part. Do not forget. Okay? Everybody is going to do the summary part.
Bye.
Hello.